Hello everyone. Today we'll be discussing clinical epidemiology, the meta-analysis. To begin, we first need to understand what a meta-analysis is. A meta-analysis is a statistical tool or strategy that uses previous independent research to estimate or calculate the overall variance or mean. There are 5 main steps for the meta-analysis. The first step is to determine a research question or a hypothesis. The second step is to do a systematic review. During this step, an individual will collect previous independent literature that also addresses the research question while avoiding publication biases. The third step is data extraction. During this step, an individual will extract data from each study, including data variability and various sample sizes. The fourth step is weighting the studies and standardization. Standardizing the studies will allow for comparable results and allow for one to calculate the mean and variance from all the studies. This is also referred to as the effect sizes. The last step is the final estimate. The final estimate compares effect sizes between the independent literatures. Common models to compare effect sizes are random and fixed effects.
Summary intervention effect estimate is calculated using a weighted average of intervention effects estimated by individual studies, where Yi is the intervention effect estimated in the ith study, Wi is the weight given to the ith study, and the summation is across all studies. If all the weights are the same, then the weighted average is equal to the mean intervention effect. The bigger the weight given to the ith study, the more it will contribute to the weighted average. The standard error can then be derived to find the confidence interval, demonstrating the uncertainty of the estimate. P-value can also be derived, communicating the strength of the evidence against the null hypothesis. The intervention effect estimates across the studies may use a random effects model, whereby it is assumed that studies are not estimating the same intervention, but instead a distribution across studies. Alternatively, if it is assumed that each study is estimating the same quantity, then this is called the fixed effect meta-analysis. It is also important to assess the variation among the results of separate studies and whether it consists with random variation to address biases. Meta-analyses are usually demonstrated using a forest plot. A forest plot displays both effect estimates and confidence intervals for both individual studies and the meta-analysis using a block surrounding the point of estimate. The area of the block also indicates the weight assigned to that study within the meta-analysis, with larger blocks representing more heavily weighted studies in the meta-analysis.
Although the potential in using the meta-analysis is very promising, it is important to understand some of the limitations associated with the technique. One of the major flaws associated with meta-analysis is heterogeneity. Since meta-analysis combines different studies with varying methodology, population, and other factors, this may affect the reliability of the results. Another limitation of meta-analysis is that they are dependent on the data available from previous studies. That being said, if the studies contain flaws in its data collection or analysis, the meta-analysis will not be reliable. Lastly, a meta-analysis is able to identify association between variables, but fails to establish causation. This is because the technique fails to look at other factors that may account for a trend and rather focuses on statistical significance. Although the results may be statistically significant, they may be small and not as clinically significant.
Before diving into explaining how meta-analysis can be used in evidence-based practice, it's important to understand what exactly this type of practice is. This practice refers to a problem-solving and decision-making approach which involves and relies heavily on scientific research. It's a process in which scientific research is reviewed, analyzed, and transitioned into creating clinical practices that are best suitable for the individual or patient. Evidence-based practice takes scientific evidence, clinical experience, and patient values all into consideration when trying to form and create a clinical practice plan. In regards to clinical experience, it refers to the knowledge and skills the experts themselves have, while patient values refers to a set of personal circumstances and priorities the client identifies And scientific research is the most accurate and concrete information that's gathered from scientific literature to support a plan. The scientific research element that goes into evidence-based practice is where meta-analysis can be used in a beneficial manner. As previously mentioned, meta-analysis is a statistical procedure that combines multiple studies into one by investigating common problems or issues and then evaluating the efficacy of interventions across various studies. Many clinicians may not have time to evaluate multiple studies when trying to create practices, so meta-analysis can be used to pull together outcomes from multiple studies to create and integrate empirical research when deciding on the best practice possible. Therefore, it allows researchers to use compiled data and literature to come up with the best optimal solution while looking at similarities, differences, and trends across various studies.